Thank you very much. Well, I'm Roger Highfield. I'm the Science Director of the Science Museum. Great pleasure to be here with Venki to talk about his wonderful book. Sorry, we must present it properly here. Do I need to use the microphone, or can you all hear it? Sorry about that. Okay, Roger Highfield, Science Director, Science Museum, and we have Sir Venki Ramakrishnan of the MRC's Laboratory of Molecular Biology, President of the Royal Society, and of course the winner of the 2009 Nobel Prize. And I suppose the thing I'd like to... Yes, indeed. And here's the amazing hoopla in Stockholm. And I suppose, you know, the way I'd like to structure this conversation, Venki, is to start off with the young Venki and take us here with all the kind of trials, tribulations, the competition. We'll talk a little bit about the prizes and so on. But I'm sure everyone in the audience wants to know, you know, was there anything in your childhood or your upbringing that gave you, you know, did you, at the age of 4, were you shouting, Nobel, Nobel, or something? You know, what helped get you into science?
I think, well, my parents were scientists, so I was exposed to science from quite an early age. But I actually wasn't that interested in science. Oddly enough, I was more interested in English and and just goofing off. And, but then
But goofing off with a purpose? Were you studying things or really goofing off?
I read a lot, but nothing to do with my curriculum. And I think I was influenced by a good science teacher in my last 2 or 3 years of high school. And then I decided to become a physics major for my undergraduate, and that really sort of set my path on to science. But I have to say, even when I eventually, as you know, I quit physics right after my PhD.
Well, I was going to say, you kind of got off to a false start in science, didn't you? In the book, you described how you were a physicist first.
Yeah, I was 19 when I graduated and went to the US to get a PhD in physics. And I found I wasn't really interested in physics. Once I finished all my coursework and started doing research, I thought, well...
Yeah, I think you just have to be a...
Yeah. So once I finished all my coursework and actually had to start working, trying to be original in physics, I didn't find it very engaging. And I thought I would be better off starting over again and becoming a biologist. And so I actually went back to graduate school in biology. I started all over again with the option of getting a second PhD. So I would say my career in science was not at all smooth. And, you know, for the longest time, I was not in what you would call the fast track main centers of science. You know, science also sort of seems to segregate in clusters. You know, in Britain, you have, you know, your Cambridge and London and Oxford and, you know, other places like Edinburgh and so on. And in the US, it's largely concentrated in the Northeast and in California. And I wasn't in one of those places. I went to a small sort of Midwestern university in Ohio. And then when I switched to biology, I went to University of California. And then after 2 years of that, I decided I was ready for a postdoc, and I went to Yale. That was my first experience with, you know, a high profile center of research. And I have to say I suffered from a kind of imposter syndrome because I'd gone to my local state university in India, and then I'd gone to Ohio University, and then did this slightly odd transition.
In fact, how did you because you were in Tamil Nadu, and then how did you end up in, you know, why the States? Because there obviously are lots of opportunities all over the planet.
Well, you know, it's funny, when my father was a young man, the default destination for Indians was to go to Britain, you know, ideally to Cambridge or someplace like that. But
well, they got you in the end, didn't they?
But by the time, yes, that's true. But by 1971, which isn't that much after independence, which was 1945, most of my cohort never even considered going to any part of Europe, including Britain. You know, most of them, the immediate thing was, if you want to go abroad, you go to the United States. And that, I think, has changed somewhat in the last 40 years. Because there's been a resurgence of European science, and I talk about this as part of, you know, the EU has actually really encouraged European science in many ways, and it's led to a resurgence of European science. So now Europe is also a destination, including Britain.
We can talk about you-know-what perhaps at the end if we've got time. We all know what I'm talking about there. But obviously the gene machine is about this amazing machine that's in all your bodies, the ribosome. It's turning genes into flesh and blood. What was it that, you know, when did you first get a sense that that was what you were going to set your sights on?
Well, you know, the funny thing is, everybody thinks they know what genes are. We could discuss that later if you want. But actually, they don't. You know, if I ask you, do you know what genes are? Most of you will say, yes, we know what genes are. But if I probe you even a little bit, you'll be lost very quickly. Okay, you won't actually know what a gene is. And I didn't know that genes are essentially codes of information that contain instructions on how to build life through making proteins, through making lots of different proteins which make up life. And the machine that reads genes to make proteins is called the ribosome. And like nearly all of you, I hadn't, even after my PhD in physics, I'd never heard of the ribosome. And then I went to the University of California, San Diego to start studying biology, and I realized how important the ribosome was. And after a year and a half, I realized I knew quite a bit of biology by now. I knew how to do lab work, and I didn't really need a second PhD. And so I came across an article in Scientific American about the ribosome, but about using a physics technique to look at the ribosome. By then, I knew how important the ribosome was and how complicated it was. And oddly enough, one of the people who had written that article was a professor named Don Engelman at Yale. And he was a guy who had offered me a postdoc straight out of my PhD, because Yale had said, we won't take you as a graduate student because you already have a PhD, but maybe some of our faculty might offer you a postdoc, and he was one of them. The other was Tom Stites. Oddly, I ended up sharing the Nobel Prize with him. But I turned them both down, saying, look, I actually don't know enough biology to do a postdoc, and sorry, I'm not interested. So when I came across this article, I wrote to Don and I said, you know, you offered me a postdoc when I didn't know a damn thing about biology. So now I actually know some stuff, so maybe you're still interested. And he said, yes, I'm going to ask my collaborator, Peter Moore, to write to you. And Peter Moore was the ribosome end of that collaboration. And he had learned about ribosomes from his PhD with Jim Watson. Now, Jim is known mainly for the structure of DNA and also for making outrageous statements, you know, to the public that constantly gets him into deeper and deeper trouble.
Yes.
But the reality is, his lab also started a lot of work on ribosomes. So there's a sort of pedigree there. And anyway, I ended up working for Peter Moore, and that's how I got into ribosomes. Yeah.
We've actually got a few slides here, which I don't want this to feel like a university lecture, but I think it's really nice to give you, you know, a bit of the sort of fibre and some of the details, because actually the more the details you learn about, even if you only get a percentage of them, it just gives you an insight into what a big, interesting problem this is. So, I mean, first of all, you know, we talked about the genomes, the recipes of, when we talk about genome size, that's all the genes in a creature. And actually, interestingly, this is the estimate in 2001, Venki, and there's a surprise here, isn't there?
Yeah, the surprise is people thought there were 35 to 45,000 genes in the human genome, but we only have about 25,000 genes. And that's not very different from A. thaliana is Arabidopsis thaliana, which has 25,000. That's a weed. And, and above it is C. elegans, which has almost the same number, about 20,000. And that's a worm, okay? So we are not, in terms of the number of genes we are not more complex than either a worm or a weed. And the lesson is that biology actually doesn't care about intelligence. Biology only cares about survival, okay? And if intelligence evolved to help us survive in the savannas of Africa, you know, that's why we're smart, okay? But it doesn't mean it's essential for life.
We've got a couple more slides here.
So this is about, you know, when you ask, if I were to ask you what are genes, many of you might say vaguely, oh, well, you know, it's those things that give us our traits. You know, we inherit good or bad genes. We might have genetic defects that cause disease and so on. But what genes are instructions on how to make a protein, but also when to make it and how much of it to make. So there's a lot of fine-tuning there. So then that naturally leads to what are proteins? And proteins are long chains of building blocks called amino acids. So if you look at those little things jutting out of the chain on the top, you see they have different shapes and, you know, sizes. And that's because there are 20 of them that contain different shapes, sizes, and properties. But the interesting thing is that depending on the order of the amino acids that go to make up that long chain, that order contains in itself the information on how to fold up the chain. So proteins work because they fold up into different shapes, and that shape gives them their characteristic function, allows them to function. So you can represent the amino acids by letters, and so I've done that in the second part. You see that row of letters, that string of letters. Now imagine if I had strips of paper and I wrote down different sentences of different lengths on each strip of paper. And depending on what I wrote down, the strip of paper would know and magically fold up into a particular shape. That's how amazing it is, that just the order of those letters has the information on how to fold it up. And this, on the bottom, are 3 folded up, you know, final structures of proteins. That long filament with 3 colors is a 3-stranded helix, which is collagen. That's what makes up our skin and connective tissue and cartilage and so on. And actually, by weight, it's the most abundant protein in your body. And then this one on the bottom left is hemoglobin, and it carries oxygen from your lungs through your blood into your tissues. And that's what gives blood its characteristic red color. So red blood cells are filled with hemoglobin. They're basically bags containing hemoglobin. And on the bottom right is rhodopsin. This is a protein that sits in the membrane, which is to say the outer surface of cells in your retina, in your eyes. And it's what allows you to see, because this is a protein that detects light and sends a signal to your brain that light's been detected at this particular point. So the point of this is to show you proteins do amazing different things, and we have thousands of them, and each of them is made by carrying out instructions in our genes.
We've got that right here, just talking about, so we start off with the DNA, and then there we are, that's the
so I
4
like to think
bases
of,
come in pairs.
So since we're at the British Library, I want to make another analogy. So you can think of DNA, our genome, as a collection of all the books we might ever want to use, okay, all the information we might ever want to use. Now, if you go next door, I mean, to a different part of this building where the stacks are, and go there and say, I want to pick out this book from the 1700s, the fellow will say, sorry, we're not going to let you take that book out, but we'll give you a photocopy or a digital scan of the book, and you can read it. Okay, so it's very similar to that. The cell has all of its information as DNA, but as it needs each gene, it copies a section of the DNA into RNA, and then that's read But now you have to go from a language of genes, which is a 4-letter alphabet, because DNA and this copy called RNA have only 4 types of building blocks. And proteins, as you know, have 20 types of amino acids. So it's like going from a 4-letter alphabet to a 20-letter alphabet. And that's why the process is called translation. And the way the cell solves this problem is by reading units of 3 bases on RNA, and each of those 3 bases corresponds to a building block of proteins called amino acids. And that is done by an adapter molecule, which if you click, is one that brings in the right amino acid, but at the other end has a group of 3 bases that sort of locks in with the 3 bases. No, that's fine. Now the point is it doesn't happen by itself, and it happens in these particles called ribosomes, which were first discovered in the cell as these blobs where newly made proteins were localized. And then if you expand again, these blobs were isolated, and they were all, if you look at them, they all have a large and a small part called the large and small subunit. And so somehow this particle is where the action of reading the gene and making the proteins occurs. And if you go on one more. So if you really want to understand how that works, it's a bit like understanding any other machine. Now, I've made the analogy with a car. If you just looked at a car from a distance, you would have no idea how it worked. Okay, you just simply know that it consumed gasoline and emitted, carbon dioxide and water. If you then get closer and start looking at the organization of a car, you might have a better understanding. But it's only when you look at a car in great detail and see how an engine is put together and how it actually works and how it transmits its force through a crankshaft to the wheels and so on, and how there's steering and there's brakes, then you start to understand how a car works. And it's been the same with ever since we've known about molecules. If you know what a molecule looks like, you have you're halfway to understanding how it works.
And of course, you mentioned Jim Watson. He was famous with Crick and Franklin and all the rest of them, Wilkins, in using X-rays to get the structure of DNA. You can't focus X-rays, so you make a crystal of material, you put them through, and we're just going to talk a little bit about how that would work with the ribosome. Because
so I talk about, normally you can look at a car with your naked eyes, and that's not a problem. If you want to look at a cell, you would need a microscope. But molecules are even smaller than cells. So the point is, how would you see very small objects? So normally if you have a small object and you want to see it, you would put a lens in front of it, okay? And what the lens would do is it would gather scattered rays and condense them and recombine them into an image, okay? Now you have to realize whether the lens is there or not, the information to make the image is there in the scattered rays. So for example, all of you are looking at the screen, and the scattered rays from the screen are being combined by a lens in your eyes to form an image in your retina. And that's how you're able to see. But I notice there are quite a few empty seats here, and there are scattered rays going to those empty seats. There is no image because there's no person with a lens to recombine them, but the information is still there, okay? So that's what you have to realize. The information is there in the scattered rays, whether there's a lens or not. Now the problem with molecules is they're too small to see with a microscope, with any kind of lens. And that has to do with the fact that light, the wavelength of light, is much larger than the distance between the atoms in a molecule. And so for molecules, if you go to the next thing, we seem to be missing a slide about X-rays. So anyway.
Sorry.
That's okay. So the way...
You can sort of illustrate it a bit, can't you, Venki?
So the way you do that for molecules is you coax your molecule into forming crystals, and then you hit it with X-rays, and then the X-rays are scattered, but there's no lens for X-rays. So what you do is, in a computer, you recombine the scattered rays, doing exactly what a lens does in practice. And then you get a computer-generated image of the molecule. So to do that, you need crystals of the ribosome. And that itself is part of the story about how crystals were obtained and how it became a mad competition, to decipher the structure.
So let's move to the competition, because it was about the mid-1990s, wasn't it, when you...
Yeah.
And, of course, just to make a crystal of a ribosome is not a trivial task at all.
No. So, for example, if some of the early things to be solved were things like common salt. I mean, that was the first molecule to be solved. And you can just dissolve salt in your home and let it dry out, and you'll get crystals of common salt. Actually, it comes as little crystals. So you don't need any expertise to make crystals of common salt. But the larger the molecule gets, the harder it is to coax the molecule into very regular arrays, which is what a crystal is. It's a regular three-dimensional array of molecules. And the ribosome has 500,000 atoms. And so the idea that it would even form crystals was not absolutely certain. And the first people to crystallize the ribosome was when an Israeli scientist, Ada Yonat, went to work with a famous ribosome scientist named Heinz Gunther Wittmann in Berlin. And the two of them decided to give crystallization of ribosomes a shot. And that sort of worked. And then 5 years later, a group they crystallized the large subunit, the large half of the ribosome. And a group in Russia crystallized the small half and the entire ribosome a few years later. But then, 15 years later, there was still no progress towards an actual structure of the ribosome. And, after two rather, sort of seminal talks, seminal in the sense they changed the field in the mid-90s, many of us realized or thought that the field was stalled, and unless we and others, entered the field, nothing was going to happen. And so that was essentially what happened in the mid-90s. And the Yale group decided to take the crystals of the large subunit that Ada Yonat was focusing on, and decided that they would reproduce those crystals and start working on it. And I didn't want to do that because I thought, let them fight it out. I didn't really need, I didn't feel I wanted to compete with this large group, that was well set up. And I just started a lab in Utah with 2 graduate students. So I said, well, there weren't any good crystals of the small subunit or the entire ribosome. There were crystals, but they weren't capable of giving a high resolution, a detailed structure. And so I thought, well, let me work on the small subunit and get somewhere with that.
And I suppose we should emphasize, Venki, that people like to think of science as a quest for objective truth, and everyone's very kind of gentlemanly and polite. But when it gets like this, and there's a sniff of a Nobel Prize around, it gets pretty ugly, doesn't it?
Well, there's no sniff yet. Okay, the sniff happens once you've actually cracked the problem. Then you get a disease I call PreNoble Idea. This is a disease that afflicts scientists who've done something important, and often. People think that it might be worth a Nobel Prize, but many more people think that what they've done is worth a Nobel Prize than other people do, okay? This is this sort of self-delusion. But anyway, they suffer from this disease called PreNoble Idea. They become anxious, they want recognition. Every October, they get a little nervous in the first week of October, etc.
That's when grad students practice their Swedish accents and ring you up and sort of cause you palpitations and so on.
Anyway,
famous occurrence.
But this is after I started focusing on the 30S. I found that Ada had switched from the 50S from the large subunit to the small subunit. So she was now my direct competitor. And then Harry Noller hired the two of the Russians who worked on the crystallization of the whole ribosome. And so he started working on the whole ribosome. And of course, if they solved the whole ribosome, the small subunit was part of the whole ribosome. So in effect, he became a competitor as well. And although the Yale group was working on the large subunit, so they weren't directly overlapping, still you wanted to be the first person. You didn't want to be too far behind the other, because then it would be slightly anticlimactic. So suddenly, there was this four-way race, and the odd thing I did, the one thing you mustn't do in the middle of a tight race is actually to move, because that'll cost you a lot of time. And not only did I move from Utah, but I moved to a different continent. I moved to Britain, to Cambridge, where I work now. And there are lots of reasons for that. We could go into why I moved to Cambridge in the middle of a tight race. And luckily, it didn't actually cost me much time.
You did move to the laboratory of molecular biology, which some people jokingly call a Nobel Prize factory. So, I mean, it's got the most amazing... I mean, in sort of 30 seconds, can you sum up the culture of the LMB? There's something very special there,
yeah,
indeed.
So the LMB, it's a place which Max Perutz started with John Kendrew and others. And Max was Francis Crick's PhD supervisor, and Kendrew was Jim Watson's postdoctoral advisor. So the very year that the LMB was formed, 1962, all four of them won a Nobel Prize. So it was off to a big bang. And it's won... it has produced 17 Nobel laureates, including the last 2 years. And I don't know if we'll make a hat trick. It's very unlikely, but it's possible. But anyway, the reason that it is so successful is because it's a lab that focuses on working on really long-term important problems in small groups. I think both the small group and the important problems are an essential ingredient. The other is that it has no sense of hierarchy. All of the instruments, including 5 million pound microscopes, are open to everybody from a first-year graduate student to the lab director. And if some first-year graduate student signs up before Richard Henderson, who won the Nobel Prize for the technique, then that first-year graduate student cannot be bumped off by Richard Henderson unless he agrees to it in a friendly way, okay? So that's the kind of hierarchy, and there's no senior common room or faculty club, nothing like that. Everybody sits in the same canteen. There are no special tables for anybody, and so you'll find, first-year students sitting next to the lab director or the head of division.
Now, of course, you're portraying it as a sort of collectivist utopia, but that doesn't mean to say that you weren't hugely competitive. And there's a lovely bit in the book where you talk about a book that came out called A Milestone in Ribosome Crystallography. What was your reaction as one of the collective to that?
So of course, the LMB's view is we're very collegial within the lab, but of course we're, quite fiercely competitive with our competitors outside, in the rest of the world. And what Roger is alluding to is after a talk in 1995 that Ada Jonat gave, most of us felt that she was sort of completely lost at the time and that other people would have to get into it. And because she showed us what was supposed to be a 7 angstrom map, which at the 7 angstroms implies a certain level of detail. And we saw no features in those maps that would have that kind of detail. And these talks are usually published as proceedings of a conference. And her chapter was titled by herself as A Milestone in Ribosome Crystallography. We can leave aside the question of why someone would call their own work a milestone. I think that's for other people to say, but let's leave that aside. But I write in the book that I thought it was probably better described as a millstone because we felt, that some of her ideas of how to approach it were actually misguided. And it turned out that there were some fundamental errors, for example, in what crystallographers call the space group, which is the actual arrangement of the molecules in the crystal. And so anyway, so this is just Roger's way of saying that the book is quite frank
yes,
and doesn't pull any punches.
Now we've got about...
insofar as the lawyers would allow me.
I should say I'd love to get some questions from the audience in about 10 minutes. So we're going to try and just rattle through a couple of the... I mean, just to point out, you know, we've talked about making these crystals, and of course, you know, Venki and his team were investigating the atomic domain, but actually the sources of X-rays to do it are these amazing facilities, aren't they, Venki? I mean, this is where you get the purest and the most powerful sources of X-rays.
Yeah. So ribosome crystals, because the molecules are so large, the crystals don't scatter X-rays very well. And in order to get enough signal, you need to hit them with a very, very powerful beam of X-rays. Now, you're familiar with X-rays from your dentist, or possibly some of you have had an X-ray for a fracture in a hospital. That's a very low dose of X-rays. You can get a higher dose from an X-ray machine in a lab that we use to hit crystals with. But even that is not very strong. And if you want a very, very intense beam of X-rays, you have to go to these giant particle accelerators, which emit intense beams of X-rays.
I should say the top right image, if you see that grey thing, that's the nuclear reactor where I used to bounce neutrons off soap bubbles, just out of interest. But anyway, let's move on to the actual... So this is really the, you know, after all your hard work, Venki, this is the work that really led to the Nobel Prize, isn't it?
Yeah, so this is the complete atomic structure, shows you the arrangement of every one of those 500,000 atoms in the large and the small subunit of the ribosome. So we did the small subunit while the Yale group did the large subunit. And if you want to understand how the machine divides its labor, the small subunit is responsible for reading the code on the gene very accurately by bringing in just the right adapter molecules that bring in the tRNA, which have a perfect match to the three bases that form the code. And the large subunit is where the amino acids that are brought are stitched together to make up the protein. So there's a kind of division of labor between the small and large subunit. I like to joke that the small subunit is sort of the brains of it because it does the decoding, and the large subunit is the sort of workhorse.
And you've got this intriguing line, a glimpse at an early RNA world. And I suppose just to give a tiny bit of background there, what's fascinating about the ribosome is, you know, most of the machines in your body, or a lot of the machines, are just pure protein. But this is a mixture of RNA and protein. And there is a theory, isn't there, Venki, that the very first life on Earth did not replicate DNA, but it replicated RNA. So they talk about the RNA world. So we think this machine has been working for, what, 4 billion years or something like that?
So the ribosome was a puzzle because a ribosome is the protein-making machine in the body, but itself is made of lots of different proteins. So you can see in gray are shown all the proteins on the outside and on the left, and here you can see proteins in different colors. It's a little more complex. I would have shown a different slide for that, but the main point is...
We've got another one. There's
oh, well, this is the entire ribosome with about 500,000 atoms. But anyway, the point was, Crick thought, well, this is a puzzle. It's a chicken or egg puzzle, because if the ribosome, which makes proteins, is itself made up of proteins, where did the ribosome even come from? And he suggested that, well, actually the early ribosome might actually have been made entirely of RNA. At the time, people didn't take him seriously because there was no example of RNA being able to do anything. It was either thought to be an inert scaffold, or it was thought to be a carrier of genetic information, like a copy of DNA. And it's only when in the 80s, people discovered that there were RNA molecules that could carry out chemical reactions. So that was like proteins, which can carry out lots of different chemical reactions. And so then people realized, well, if RNA can carry out reactions and assume different structures and can also carry genetic information, then in one molecule you have all of the essential ingredients of life. You have genetic information, but you also have the ability to carry out reactions and form structures. And so that led to a hypothesis that maybe life began with a world of RNA, where RNA, all the genes existed as RNA, but then RNA could also carry out the reactions of life. And gradually, the functions of RNA were replaced. So the genetic functions of RNA were replaced by DNA, and the chemical functions of life were replaced mostly by proteins. But there's still a few relics, like the ribosome, which still are largely RNA and carry out important functions.
And of course, you might think, gosh, this is all getting terribly arcane, and why are we learning all this stuff? Why are we going to all this detail? There is a very practical insight that you get out of this, because the world's actually facing a crisis at the moment in that we're running out of usable antibiotics. Superbug resistance is rising. And so, Venki, just talk us through a little bit about how understanding the ribosome can give us new insights into making antibiotics.
So it turns out about half of known antibiotics target the bacterial ribosome. And many of them are useful clinical antibiotics. And once you've solved the structure of the ribosome, it's very trivial to soak antibiotics into the ribosome, into crystals of the ribosome, carry out the whole experiment again, and take a calculated difference. And the difference will tell you where the antibiotics are. And so, for example, we could see a blob for this antibiotic called spectinomycin, and it's present in a little crevice, if you look on the right, and you see those little atoms there. It's present in a little tiny crevice in the ribosome. And so just a small molecule, by binding to a critical pocket, is able to stop this entire machine from working. And using that, we were able to solve lots of different antibiotic structures. And of course,
I think we might have another
so here
one
are
as well. Yeah.
6 of them. And the Yale group and Ada Yonat have solved many others. And so drug companies are now trying to design better antibiotics by using these structures. For example, antibiotics that will bind to the same pockets but might be less toxic and maybe more effective against resistant organisms.
I think just finally, before we open up to questions, and I can see I've got the very first one there, let's see a film of the ribosome in action, because I think what's fascinating is these are all very static images. In your body, you've got these soft, deformable molecular machines doing the most amazing tasks. I just hope this video will work. And Venki, you better give the commentary to it, because, sorry, I'm skipping a little bit, the Nobel Prize. Do you want to talk? You've got some misgivings, I should say, about the Nobel Prize. So we'll quickly talk about this first.
It may sound a little hypocritical because, I accepted.
Come on, you
prizes.
can say that now that you've got one, Venki. Yeah. I mean,
but I can tell you
now he says it. Okay.
Now I can say it, but I couldn't say it before, because then you would accuse me of having sour grapes.
Right.
Okay? And so that's a problem. If you complain about prizes when you don't have one, people think you just have sour grapes. Okay, so the right time is, you've got to show if you have it and you complain about it, you can be taken a bit more seriously. The problem with prizes is that they convert science to a kind of sports metaphor of, first, second, and third, etc. Now, in a 100-meter race, you can, first of all, the rules are very well defined, and secondly, measuring the outcome is very well defined. You can clearly measure who came first, second, and third. But in science, different people contribute different bits of the puzzle towards a problem, or they contribute to different steps along the way. They make something possible, which then eventually leads to some breakthrough. And so deciding which were the really important contributions isn't so clear. And the other is that science isn't limited to 3 people, you know, or at most 3 people. And often more than 3 people contribute to a prize. So those are 2 problems generally with prizes.
There, we've got
And
this
for
nice
example,
slide
here's
to
my
show
team. You know, these are the people who worked on the ribosome just in my lab. And, you know, of course, every other lab had things like that, people like that.
And only 3 people can win the Nobel Prize.
Right,
so
and often only the head of the lab. I mean, you could argue that I was the one setting direction for my lab, and these people came to work under my sort of direction. So there's often the skipper versus crew analogy that's been used. It was notably used by Anthony Hewish, who got the prize that Jocelyn Bell didn't, you know, get. Okay, so that is a point of view. Anyway, but what I want to say was that there's the rule of 3. There is also the thing about that's specific about the Nobel that I want to, you know, disburden you of this notion, which is that many people think that if you're a Nobel laureate, you're some sort of genius. And the reality is the Nobel Prize is given for making an important advance or an important discovery. And people can do that in lots of different ways. You know, I point out, Shakespeare said, some are born great and some achieve greatness and some have greatness thrust upon them. And the same is true of Nobel Prizes. You know, sometimes you sometimes you are very smart and you have an insight that other people don't. Other times you're just well set up and you're very persistent and you're in the right place where you can carry through that determination. And other times you simply stumble onto a discovery and you know, are lucky enough to realize what its importance is.
There is a great quote on page 237. This is the amazing day when Venki got the Swedish voice on the other end of the phone, established that it was not a graduate student playing a trick. It was indeed the real deal, and his wife Vera said, I thought you had to be really smart to win one of those.
Yes. And who should know better than she, right?
So I think let's look at I hope this is the movie now, because you'll suddenly get a vivid insight into what's happening in your cells. So here we go.
So this is just a picture of the ribosome in action. And it all starts off with the small subunit of the ribosome binding that long snaky molecule, which is our gene, or the messenger RNA carrying the gene. And it figures out where to start, and the blue molecule is the adapter that's brought in the first amino acid. And then the large subunit joins, and then these little other blobs are protein factors that help the ribosome figure out exactly where to start, and they leave. And once they've left, then new amino acids are brought into the ribosome, but they're brought in by those adapter molecules, but those adapter molecules also have a chaperone, which is this little yellow thing which is a protein. And if it's the right molecule, right tRNA, or the right adapter, corresponding to the code, the ribosome accepts it. And then you can't see it because it's buried in the small subunit. The amino acids brought are joined. And then the whole thing has to move so that the next bit of code can be read. So that happens in a complicated process. You can see how the mRNA has been dragged over and the 2 adapter molecules have moved over a slot. So now the original slot is open again for the next molecule. And so you can see how adapter molecules bring in amino acids to the ribosome, and the code is being read, and the protein chain, as it's made, comes out of a tunnel and then emerges, and then it has in itself the information on how to fold up. And so you can see it's gradually folding up into a 3-dimensional structure as it comes out of the ribosome. And this continues until there's a special signal which tells a cell when to stop, when it's reached the end of the gene. And when it reaches that special signal, a particular protein binds to the ribosome and cleaves off the newly made protein so it can go off and do its thing. And then there are other things that come and bind to the ribosome that split it apart, so the whole thing can start over again. And while you've been listening to us, in the trillions of cells in your body, there have been thousands, each of them, there are thousands of ribosomes making thousands and thousands of proteins from your sort of thousands of genes. So it's amazing to know that it's a process that goes on all the time, and life wouldn't be possible without it.
Well, I hope your minds are boggled. Let's have a couple of questions. There's a very persistent hand going up here, so fire away with the first question.
It was a brilliant presentation, and thank you for the session. Quick question is, would this affect aging? And what do you have to say about this whole thing about stem cells and being able to re-engineer DNAs and live up to, maybe you want to live up to 200 years and look very young, etc.?
Yes. So the aging has to do with lots of things. So if you think of life, it's a very well-regulated, very precisely regulated orchestration of how genes are expressed at different times, in different cells, and so on. And as we age, two things happen. The energy, all of life requires energy, and the organelle that is responsible for creating molecules of high energy in our cells are called mitochondria, and they suffer damage. So that is one problem. Another is that our genes themselves get damaged. We have elaborate mechanisms to repair damage, because damage is occurring all the time. Thousands and thousands of DNA damage events in cells every hour, but they're constantly being repaired. But at some point, the damage sort of escapes the repair mechanism. So you get an accumulation of damage. That leads to a loss of regulation of genes. And that in turn means that the program is slowly going awry. We're making proteins that have defects, or we're making too much of them or not enough of them. And that's ultimately leads to aging.
And obviously, if you understand this, I guess we can come up with anti-aging. That's really where this is heading. Come on, Venki, we want the anti-aging pill.
So it's oddly enough, my publisher asked me whether I wanted to write any other books. And one of the ideas I have is a book on death and aging, why we die at all, and why some species seem to have extremely long lifespans.
It's a great subject. There's another question over there and more in the front. I'll do my best to fit everyone in the next 10 minutes. If you run like the wind, there we are. Fire away.
Oh, hello. Amazing talk. I want to know, there is at the moment a huge interest in the use of artificial intelligence in these fields, and there is a feeling that sometimes this is how much do you think this is like the emperor's new clothes? And how it's actually where it is actually useful, the use of artificial intelligence in
I can tell you something about nearly every new technology or field of science that leads to new technology. What happens is you get a big breakthrough in science that leads to potential new technology. There's a huge amount of hype, okay? And then people get disillusioned, and they call it the emperor's new clothes. And they go away, most of the people disappear, and then there are people who continue on in that field, and eventually there's a second wave, and that's what often causes a real revolution. That's happened many, many times. For example, recombinant DNA, there was a huge amount of hype, okay? And then nothing happened, but now we have a huge biotech industry based on recombinant DNA technology. It's the same with artificial intelligence. The first ideas of artificial intelligence were strictly by coding every possible eventuality and trying to anticipate how a machine might think. And then, people like Geoffrey Hinton, who's actually here in London before he moved to Toronto, thought of the idea of deep neural networks and unsupervised reinforcement learning, which is a bit like how a child learns. A child doesn't learn by thinking through what it needs to do. It learns by trial and error and figuring out what works and what doesn't. And that's how possibly children also learn language. And so anyway, that has really taken off. And so to the point where artificial intelligence today is becoming increasingly powerful. And I would expect that it's going to make huge inroads into helping scientists solve problems, including problems in biology.
But there is a lot of hype, you're right. There's another question just here, and then one in the front here, and another one in the front, far away.
You made the whole journey sound as though it was a cakewalk. I'm sure you had so many difficulties, especially coming from Tamil Nadu like me. Now, all I want to ask you is, how did you manage when you hit a wall, especially in the lab, as a pretend researcher in surgery? I just want to know how you coped with the frustrations and the timelines that you had during your research days.
So, as you know, I had a number of what I would call failures in my life. Firstly, I went to, in India, all of the bright students take this horrible entrance exam for which they're preparing for about 3 years to get into a few schools, engineering schools, or they go on and become doctors. And I didn't get into one of these IITs because, my parents had the view that you don't take these coaching classes. If you're smart, you'll get in. And I didn't get in. So what I didn't realize is that all the people who take these exams and get in have been putting huge amounts of hours of effort into overcoming these, just to prepare for these entrance exams. Anyway, but I got a National Science Talent Scholarship to do basic science. I went to my local state university because my parents decided I was too young. I was 15, too young to go off. They thought I might get into bad habits if I went off to Delhi or some well-known university. So I went to my local state university, and then I went to Ohio University in the US, and at the end of it, I realized I wasn't cut out to be a physicist. So here I was, I'd spent all this time, and I was going to have to start all over again. Luckily, I had just gotten married, and I had a 1-month-old child and a 6-year-old stepdaughter at the time. And luckily, my wife was okay with my going back to graduate school and starting over again. Then I go to graduate school. I didn't even understand what the professors were talking about in my first week. And I realized I had to take undergraduate classes, to catch up. So that was, another setback. Then I went to work for a postdoc, and at the end of my postdoc, I applied to 50 jobs and got 0 interviews. And I could see why. I was applying this physical technique to try and attack the ribosome. It was, in hindsight, completely inappropriate. There was a technique called neutron scattering, which Roger knows about. I too used that reactor in Grenoble. And,
Much better than Xrays.
Oh, they're terrible. It was a completely, peripheral technique in biology. And so, of course, universities didn't want to hire me, So in hindsight, I don't blame them. And so then I had to, and then I got a disastrous job in Oak Ridge that I had to escape from after a year. So finally, when I was at Brookhaven National Lab, I slowly found my feet and got into X-ray crystallography. I went on sabbatical to Cambridge and then came back. And that's when my life really sort of got on track, if you like, in terms of doing really first-rate science. But it was quite a long slog, and I could have fallen off the edge, and I point that out in the book. The book is very, very frank about all of the sort of bad decisions I made and how I recovered from them. And, so, yes, you're right. It wasn't a straightforward journey.
Time for just a couple of very quick questions. There's one here, and then the last chat. But better be a good question, okay? But be really quick.
Right, really quick. I understand that this protein manufacturing process leads to the basic structure of tissues, such as nervous tissue. Am I right in thinking that the slightly clunky-looking protein manufacturing thing has nothing to do with something as sensitive as a thought, which is more of an electron-electrical kind of a thing? I would not like to think my thoughts were made up of clunky things.
I hate to tell you, but nearly all your thoughts are enabled by these clunky proteins.
Yeah, enabled. That's right. That's the key thing.
Yes,
Let's
well, you have to ask, what is there beyond, signals being transmitted, from neuron to neuron, and also memory. The preservation of memory is made possible by proteins.
The transfer of signals is not proteins, is it?
No, it's an electrical signal. Quite right. It's an electrical signal, but the thing that makes the electrical signal possible are proteins that allow certain charges to go through and other charges not to go through. So it's all done by proteins in the end.
So we've got one question here, and then the very last one there.
Can I just ask you about gene therapies and neurodegenerative illnesses, which are mainly protein misfolding diseases? Are we kind of really having a problem with the technology or with the basic knowledge that we can't crack the code really for these things?
No, I think the technology is rapidly being developed, and I would say people are using it in animals already. There are two questions with using it in humans. One is safety, to make sure that when you modify a gene, you've done no other alteration, and also that correcting the gene is not harmful in itself. Okay, so that's one problem of safety. The other problem is an ethical problem of when to allow it and for what to allow it. So for example, somebody did gene modification on these babies in China by doing gene editing on their embryos, and it was a completely stupid application of the technique, and nobody's heard of him. My suspicion is that he's probably in jail, and since that happened.
I should say there's some amazing gene therapies for blindness developed in the UK, and the UK has done some really remarkable work here. But just the very last question here, no pressure, but it's got to be an amazing question, okay?
I'm going to preface this by saying how amazing this talk was. I'm just bowled over. My question is this, and it goes back to your roots in India and then in the West. You made what I think is a very profound statement, where in a collegiate atmosphere, people are treated fairly and equally, regardless of regardless whether they're a first graduate or a Nobel Prize piece. Can I just, before I come to the question, I just want to give you this scenario.
Come on, it's got to be a quick question.
I just need to say this,
Okay.
and I'll give the question. In India, if you just go to the question is the state of science in India, but before I get to that, just turning the table slightly. If you take, for example, as an example, the judicial system in India, you go along the galleries of where the judges walk and interact with the public. And now everyone is supposed to bow down to the judge and be in awe and reverence and so on. And if you don't do that, a judicial officer, lower down in the scale, will be sacked. And that's a protocol which has just been issued. I want to know your views on that, because you've walked the path. You're talking about equality, and you're talking about this fantastic equality between undergraduates and postgraduates and postdoctoral people, and you're sitting there and giving an amazing science story combined with human interaction.
Yeah, so I have to say different disciplines have their own traditions. So, the thing with law is that, law is application of a code, and it has its own tradition and rigor and so on. So I can't really speak for the judicial tradition and my guess is that in a good system, you ought to be able to criticize a judge or be able to say what you think. However, in science, if you don't encourage freedom of thought and the freedom to disagree with your seniors, and in fact to disagree with big shots in the field, then you can't make progress, because science often depends on questioning current ideas and overturning them in favor of something that turns out to be correct or more correct. So I would say science really does depend absolutely on freedom of conversation and freedom of thought and expression.
In fact, the Royal Society's motto is, in effect, take nobody's word for it,
Yeah,
which I
exactly.
think sums it up perfectly. Yeah. Look, those are wonderful questions, and sadly, we've run out of time. I just want to thank you, the audience, for the great questions. Thank you to the festival for hosting us. Thank you to the Royal Society. And finally, thank you to the wonderful Venki Ramakrishnan. Thank you.